Hi everyone, we are about to talk PCR test detection of Escherichia coli O157H7. So we are the team 10 of the subject Bacteriology in the semester 2025-2 of the group 4. We are conformed by Flores Cinco Rosario Itzel, Lun Ignacio Melissa Carel, and Perez Ayala Daniel Alejandro. We're going to start talking about microscopic characteristics of Escherichia coli, which is a rod-shaped non-spore-forming bacteria. It's member of the family Enterobacteriaceae. It has an approximate size of 0.3 to 0.5 micrometers wide by 1 to 6 micrometers long, although it may vary depending on the strain. It is a gram-negative bacilli that share a common antigen known as enterobacterial common antigen. This strain expresses a somatic or cell wall antigen O157 and flagella antigen H7, and can grow rapidly aerobically and anaerobically. It means it's a facultative anaerobe on a variety of non-selective and selective media, for example, blood agar and McConkey agar, respectively. It uses a simple nutritional requirements, ferment glucose, reduce nitrate, and are catalase positive and oxidase negative. Therefore, using the Liebel and Clark's model in an E. coli O157H7 infection, which focuses on the disease progression and prevention opportunities, we can map how the infection progresses and where interventions can be targeted. In the prepathogenesis period, the susceptibility is higher in children, elderly, or immunocompromised individuals. Also, with the ingest of contaminated food like undercooked beef, raw milk, unwashed vegetables, contaminated water, or poor hygiene practices in food handling. In the early pathogenesis stage, or preclinical incubation, the route of entry of the bacteria is the ingestion of contaminated food or water. Has an incubation period of typically 3 or 4 days in a range of 1 to 10 days. When the bacteria attach to the intestinal epithelium and begin producing the Shiga toxin, it's called decolonization. And there are no symptoms yet, but pathophysiological changes begin at the intestinal mucosa. For a detection opportunity, surveillance during outbreaks, early lab testing in exposed individuals. In the clinical pathogenesis stage, the initial symptoms or prodromal phase are abdominal cramps and non-bloody diarrhea, which within 1 to 2 days the diarrhea becomes bloody and it's called hemorrhagic colitis. There exist complications in the 5 to 10% of the cases, especially in children, which develop hemolytic uremic syndrome, which is a life-threatening condition involving acute kidney failure, hemolytic anemia, and thrombocytopenia. For clinical management, no antibiotics has to be used because they can worsen the toxin release, and we have to make a supportive care, like hydration and monitoring kidney function. Resolution stage, most healthy adults recover within 5 to 10 days without complications. The complications are hemolytic uremic syndromes can lead to a renal failure that require dialysis. Rarely, long-term effects include hypertension or chronic kidney disease. The mortality in this stage are rare but possible with the syndrome, within 3 to 5% of the cases. The PCR test focuses on the detection of O157H7 strain by searching Shiga toxin production or associated genes. The DNA is the genetic information that has the blueprint of the cell, while the RNA is an intermediate molecule that converts this blueprint into defined amino acid sequences into proteins. It has an intimate tier adherent system that promotes intimate attachment and colonization of intestinal epithelial cells following initial fimbrial adherence. It's a protein anchored in the outer membrane and peptidoglycan, and the tier delivers the host cell cytoplasm through the type 3 secretion system. The O antigen composes the outer polysaccharide chain linked to the core polysaccharide of the bacterial cell wall, and the H antigens are located on the flagella. The Shiga-like toxins induces ribotoxic stress response and endoplasmic reticulum stress response, and induces inflammation, apoptosis, and autophagy. It's a lysogenic phage-encoded AB5 exotoxin that disrupts protein synthesis in the epithelial cells lining the intestinal mucosa, leading to cell death, mucosal sloughin, and eventually bloody diarrhea. The plasmid O is correlated with hemolytic activity and adherence to intestinal cells. Expressa catalase peroxidase, an extracellular serine protease, has 4 genes of the hemolysis locus and 13 genes comprising a type 2 secretion system. The locus of enterocyte effacement induces the characteristic histopathological lesion known as the adhesion and effacement lesion. The adherent bacteria stimulates accumulation of host cell actin polymerization, resulting in a raised adhesion pedestal. PCR method consists of a cyclic process of 3 steps: denaturalization, primer annealing, and elongation of the DNA fragment. In the first step, double-stranded DNA is denatured into single-stranded by heating to 95 degrees Celsius. Then, specific short DNA fragments called primers are annealed to these DNA strands at 35 to 40 degrees Celsius. In the last step, primers are extended by DNA polymerase at 72 degrees Celsius by adding complementary nucleotides to 3' end to the primers. Starting from a single target DNA or RNA sequence, theoretically more than 1 billion product sequences can routinely be synthesized by a PCR in one run. So this amplification eliminates the need for extraction of large amount of pure DNA for molecular studies. A key requirement for the application of the DNA in a PCR is the availability of effective DNA polymerase enzymes. For most PCR detection applications, a previous DNA purification step is required, especially when complex matrices such as food, water, or clinical samples have to be analyzed. Many factors affect the sensitivity of PCR methods. For instance, inhibitors can act in different points of the reaction, interfering in the cell lysis, degrading nucleic acids, or inhibiting polymerase activity. Thus, DNA purification methods must provide enough removing of sample residues, additives, preservatives, and so forth to allow the proper amplification during the PCR reaction. Conventional procedures for isolation of nucleic acid involve cell lysis with lysosome, digestion of contaminating proteins with proteinase K, phenol chloroform extraction of degraded proteins and cell debris, and a final ethanol precipitation of nucleic acids. Furthermore, DNA can be extracted by alkylating resin with proteinase K digestion and phenol chloroform extraction. In the clinical ambit, stool is the one in which E. coli is more often searched. These samples carry variable amounts of inhibitory substances such as biliary salts, proteins, polysaccharide complexes, bilirubins, and phenol compounds. In the reagents, first we need the primers, which are short sequences of DNA that are designed to be complementary to regions of their gene of interest. Then we need the DNTPs, which are the nucleotides that are premixed in a preparation that contain equal quantities of all the 4 bases. Also we need magnesium chloride or magnesium sulfate because the magnesium acts as an enzyme cofactor and also impacts the specificity and stringency of primary annealing. We add reaction buffer and a nuclease-free water. And for the last one, we use a DNA polymerase that need to be thermally stable. That's why we use the Thermophilus aquaticus polymerase. But there is a number of different polymerases available. A standard thermal cycling profile starts with the naturalization for 30 to 60 seconds, the annealing for 30 to 45 seconds, and the elongation for 1 to 2 minutes. This repeats between 25 and 40 cycles, and then add a final extension step for 8 to 10 minutes of 70 degrees Celsius. A negative control is a PCR reaction with no amplification. It means no amplicon will be generated and is interpreted as a true negative. This kind of reaction checks whether there is any contamination within the reaction mix and checks if there is a primer dimer forming. A positive control are often used to verify that all the reagents are working, primer annealing temperatures are correct, extension times are sufficient, and there are no PCR inhibitor with experiments. It's also useful an internal control that can serve as an indicator for proper nucleic acid purification, reverse transcription control, normalization reference, as well as the PCR inhibition supervision. There should be a difference of maximum 12 amplification cycles between the internal control and the target gene. To verify whether your DNA extraction and/or PCR have been successful, the product can be analyzed using gel electrophoresis or spectrophotometry. Electrophoresis enables separation of DNA fragments according to their size through a gel matrix. This will give you an indicator of whether the extraction and/or reaction has worked, and also how much template DNA or PCR product you have, as well as giving an indicator of its purity. Spectrophotometry is usually reserved for quantification of structured DNA sample or RNA, rather than the analysis of PCR product. The advantages are high sensitivity, ability to test antimicrobial resistant, shown to be more cost-effective with selective use than culture and staining, increased ability to detect less common organisms such as viruses, and the disadvantages are a potentially lower specificity compared to the culture and staining, a need for narrow list of causative agents to use specific primers, the possibility of amplifying normal microbiota, becomes less cost-effective with performed with a multi-organism PCR approach, and supply cost, machinery fees, and training expenses. The cost of the test in Mexico is approximately 1,650 pesos plus IVA, with a sensitivity of 96% and a specificity of 97%.